Welcome to lecture 17, where we will be talking about ketone body formation and biosynthesis of cholesterol. Now, these two topics are important aspects concerning fat and lipid metabolism. And following this lecture, we will focus on synthesis of fatty acids. Now, ketone body formation happens under pathophysiological conditions such as diabetes. And cholesterol biosynthesis is important because by regulating the biosynthesis of cholesterol, we are able to regulate or maintain homeostasis such that proper cardiovascular health is maintained in an individual. In lecture 17, we focused on the process of beta oxidation. Now, by the process of beta oxidation, which is occurring mostly or predominantly in the mitochondria, fatty acid is converted to acetyl coenzyme A. And we have looked at the steps of beta oxidation. We have talked about how the fatty acids are transported inside the mitochondria, and we also talked about how to calculate the number of ATP molecules that will be produced from a specific fatty acid, and that is dependent on the length of the fatty acid or the number of carbon atoms present inside the fatty acid. Now, under normal conditions, the acetyl coenzyme A that is produced from beta oxidation goes into the TCA cycle, and through the TCA cycle, in combination with the electron transport chain, this acetyl coenzyme A is able to produce ATP. In order for acetyl coenzyme A to go into the TCA cycle, the metabolite that is required, and I believe by now you all know that particular metabolite is oxaloacetate. However, when the body is predominantly involved in gluconeogenesis, the levels of oxaloacetate are low. And since the levels of oxaloacetate are insufficient, the acetyl coenzyme A cannot go into the TCA cycle. So, TCA cycle in gluconeogenesis will not occur because the level of oxaloacetate inside the mitochondria is insufficient, and at that time the acetyl coenzyme A that is produced from the beta oxidation of fatty acids will be converted to ketone bodies. So, when are ketone bodies formed? If somebody asks this question, the answer is when the body is involved in gluconeogenesis, the acetyl coenzyme A produced by the beta oxidation of fatty acid will be converted to ketone bodies. And we shall see in the subsequent slides why this acetyl coenzyme A has to be converted to ketone bodies. So let us look at the formation of ketone bodies, and this is important because the basic mechanism by which the ketone bodies are formed are also important with regards to cholesterol biosynthesis, and we shall see this when we go to the second part of the lecture. So in the first step, I have two acetyl coenzyme A molecules, and these two acetyl coenzyme A molecules undergo condensation by the catalytic enzyme or by the catalytic activity of the enzyme thiolase, and this leads to the production of acetoacetyl coenzyme A. And you can see here in the process, one molecule of coenzyme A is released. Now once I have acetoacetyl coenzyme A, this particular metabolite, in the presence of acetyl coenzyme A and water, is converted to beta hydroxy beta methyl glutaryl coenzyme A, specifically known as HMG CoA. And this particular process is catalyzed by the enzyme HMG coenzyme A synthase. And this particular enzyme is considered to be the rate limiting enzyme in the process of ketogenesis or ketone body formation. So if somebody asks you which enzyme catalyzes the rate limiting step in the process of ketogenesis, then the answer to that is HMG coenzyme A synthase. Please take a note of this, because this particular concept is often asked during assessment. Now, once I have HMG coenzyme A, what happens is that there is a release of one acetyl coenzyme A molecule from HMG coenzyme A by the catalytic activity of the enzyme HMG coenzyme A lyase, and this leads to the formation of the first ketone body, acetoacetate. Now this acetoacetate has two fates. First, it will undergo an oxidation reaction, it will undergo a reduction reaction where it will be converted to beta hydroxybutyrate. The proton that is required for this reduction comes from NADH. So NADH plus H plus will be converted to NAD plus. And the conversion of acetoacetate to beta hydroxybutyrate is catalyzed by the oxidoreductase hydroxybutyrate dehydrogenase. And this leads to the formation of the second ketone body, beta hydroxybutyrate. So we had the first ketone body, acetoacetate, and after this reduction reaction we have the second ketone body, which is beta hydroxybutyrate. In the ultimate step, acetoacetate is converted by decarboxylation to acetone, and since it's a decarboxylation reaction, there is a release of carbon dioxide, and the particular reaction is catalyzed by acetoacetate decarboxylase, and this leads to the formation of the third ketone body, acetone, which is volatile in nature. So we have the three ketone bodies, acetoacetate, hydroxybutyrate, and acetone. So if somebody asks you what are the three ketone bodies and how are they formed, this is the overall reaction that is helping in the formation of these three ketone bodies: acetoacetate, beta hydroxybutyrate, and acetone. And the rate limiting enzyme that is catalyzing the formation of ketone bodies is HMG coenzyme A synthase. Together, acetone, acetoacetate, and beta hydroxybutyrate are known as ketone bodies. And you can see here that you have a ketone group present in each of the ketone body molecule or metabolite that is produced by the process of ketogenesis. Now we will try to appreciate why during gluconeogenesis the formation of ketone bodies is so important. So in gluconeogenesis, if I go back to the definition of gluconeogenesis, gluconeogenesis is the formation of glucose from non-carbohydrate sources, and that term non-carbohydrate is very, very important. So one of the non-carbohydrate sources from which glucose is derived is triacylglycerol. And how this process happens: first, in the fat cell in the adipocytes, there is lipolysis, and I mentioned this in lecture 16. By the process of lipolysis, which is a hydrolysis reaction, I get glycerol and fatty acid. These glycerol molecules and the fatty acids derived from triacylglycerol are carried by blood to the liver, or more specifically the hepatocytes, where the glycerol is converted to dihydroxyacetone phosphate, and by the process of gluconeogenesis you get glucose. The fatty acid, of course, has to go inside the mitochondria, and we talked about this in detail. Inside the mitochondria, they are converted to acetyl coenzyme A. Since there are no oxaloacetate molecules or insufficient number of oxaloacetate molecules, the excess acetyl coenzyme A that cannot go into the TCA cycle is converted to ketone bodies. These ketone bodies are released by the hepatocytes, carried by the blood, and they act as fuel predominantly for the brain, to some extent for the heart and renal cortex. So this is how ketone bodies help in the maintenance of homeostasis under conditions when the person is starving or when gluconeogenesis is happening inside the physiological environment. Now, if I want to look at this process again, under normal physiological conditions, I need to maintain the levels of glucose in the blood at 40 milligrams per deciliter. Under starvation, when the person is not having enough glucose, the body is in a gluconeogenetic mode, and in this we are breaking down triacylglycerol to glycerol and fatty acids. This glycerol is converted to dihydroxyacetone phosphate, that goes into gluconeogenesis, and through gluconeogenesis you produce glucose, which is then used by the body for the generation of ATP, and depending on the condition it may be either aerobic or anaerobic in nature. Body also breaks down amino acids to glucose, and we shall talk about this when we talk about amino acid metabolism, but predominantly this topic should be dealt when Prof. Riyad talks about integration of metabolism as well as metabolism during different feeding states. The problem is the body does not have excess store of amino acid. Therefore, if the body starts breaking down proteins to amino acids and then convert those amino acids to glucose, it will affect the metabolic pathways detrimentally. And the reason is amino acids are important constituents for the enzymes that are involved in these metabolic pathways. Therefore, the function of the whole physiological system will be impaired or detrimentally affected. So what happens is the ketone bodies that are generated from fatty acids help in sustaining the key organs, especially the brain. So under starvation or during starvation, hepatocytes are in a gluconeogenetic state. By the beta oxidation of fatty acids, they produce acetyl coenzyme A, and this acetyl coenzyme A or the excess acetyl coenzyme A are converted to ketone bodies. And as I mentioned, these ketone bodies help in sustenance of some of the key organs in the body, especially the brain. Now, why or how is the brain able to break down ketone bodies? Now, the brain expresses a particular enzyme known as thiophorase, or more specifically beta ketoacyl coenzyme A transferase. And the presence of this particular enzyme in the brain helps it to break down ketone bodies to acetyl coenzyme A. So what is the take-home message from this slide is that you should be able to tell me what is the specific enzyme that assists the brain in metabolizing ketone bodies to produce ATP. And the answer to this particular question is thiophorase or beta ketoacyl coenzyme A transferase. If you don't remember the steps, it's fine, but you should be able to remember the name of this particular enzyme. Now, once this particular enzyme helps in converting the ketone bodies, that is hydroxybutyrate, acetoacetate, to acetyl coenzyme A, then the acetyl coenzyme A that is produced will go into the TCA cycle, leading to the production of ATP through the electron transport chain. Of course, acetone is not utilized by the brain because it is volatile and therefore escapes through our breath. Now, diabetics or people who are suffering from diabetes, they have a condition known as ketoacidosis, and this particular condition is known as diabetic ketoacidosis. So let us take the two major forms of diabetes. Of course, I have not talked about gestational diabetes here, which I believe Professor Bayumi will teach you when he talks about diabetes towards the end of this course. So the first, type 1 diabetes, where the beta cells of the islets of Langerhans are destroyed and the body does not produce sufficient amount of insulin. As a result, these kind of patients, they suffered from high levels of blood glucose. In type 2, although the body produces insulin, the patient is resistant to that particular insulin for reasons which are not properly understood till date, and also the patient in type 2 diabetes has high levels of blood glucose. Now, the reason in both type 1 and type 2 the person is having high levels of blood glucose is because of the fact that the glucose is not able to enter the cells. So cells are in a virtual starving mode, and the glucose that should enter the cells which concern or which are associated with metabolism are the adipose tissue and the muscle cells. Because insulin is not produced in type 1, therefore the tyrosine kinase receptor is not activated, which does not increase the expression of the GLUT2 transporter on the cell membrane. As a result, the glucose in type 1 cannot enter the cell. In type 2, although the body produces insulin, but since the body is resistant to the insulin, also the tyrosine kinase receptor for insulin is not activated, and GLUT2 expression is unaltered, or it is practically not there. As a result, there is less entry of glucose in specific sites, especially the adipose tissue and muscle. So what happens is, since the body is virtually in a starving mode, the liver becomes stuck in a gluconeogenic state. And because of this, there is ketogenesis, and this ketogenesis leads to ketoacidosis. What you are going to tell me is that why the increase in concentration of ketone bodies leads to acidosis, and that's a take-home assignment for you, and this you should answer as a short answer question. And as I said, these assignments are formative in nature, and you may expect short answer questions from these formative assignments during your end of course assessment. So because of this virtual starvation that is happening in either type 1 or type 2 diabetes, the liver becomes stuck in the gluconeogenic and the ketogenic state, and because of the production of ketone bodies, there is ketoacidosis. Of course, part of the ketone bodies that are produced by the hepatocytes are used up by the brain for the generation of energy, and also to some extent by the cardiomyocytes and the renal cortex. So that's the end of the session for ketone bodies. Of course, there is a part related to diabetes, and I believe when Prof. Riyad talks about integration of metabolism, the different fed and fast states, as well as he talks about obesity and diabetes, he will be covering in much more detail the aspects of ketoacidosis and why in diabetes you have or what are the detrimental effects of ketoacidosis in diabetics. Now, cholesterol, before we move on into cholesterol metabolism, which is a very, very complex metabolic pathway, and I am going to make it very simple for you. We need to understand the basic structure of cholesterol. So cholesterol has a steroid nucleus, and you can see that the arrangement of the cyclic rings here is the core of the steroid nucleus here. And apart from the steroid nucleus, it contains a 8-carbon hydrocarbon tail, which is attached to this particular position here. And also we have a hydroxyl group at this particular position, which forms esters with fatty acid, and cholesterol ester is the predominant storage form of cholesterol in an individual. So what is the classic structural identity of a cholesterol molecule? The answer is the steroid nucleus. So if I give you the structure of steroid nucleus and ask you which of the following metabolites have this particular nucleus in terms of their molecular structure, you should be able to identify cholesterol. Pretty simple and straightforward MCQ that is often asked in an assessment. Cholesterol metabolism in detail will be covered in cardiovascular system. Also, I think they will talk about it when they talk about digestion of fats, because I mentioned this in the class that digestion of fats requires bile acids. So cholesterol is predominantly synthesized from acetyl coenzyme A, and it is eliminated as bile acids, and that's the reason bile acid sequestrants are often used for treating hypercholesterolemia. Of course, cholesterol has various important functions in the body, and it is the precursor for all steroid hormones, and most importantly, it is the precursor for vitamin D. So if the levels of cholesterol in an individual are severely depleted, there will be specific impairments, and the person may suffer from deficiency of vitamin D. Most of the time, it is the product of animal metabolism, because plant sterols are excreted out. We are not able to metabolize plant sterols, and that is the reason the diet of a person who is suffering from hypercholesterolemia is often, or the suggested diet should contain less amount of red meat. The predominant site of synthesis of cholesterol is the liver, but to some extent it occurs in all the tissues. So that's like some of the facts that you should remember. As I told you, the metabolism of cholesterol is quite complicated, and I wanted to make it very simple. So what I want to take home from this slide is, which of the following metabolites helps in the synthesis of cholesterol? The answer is acetyl coenzyme A. Which of the following helps in the elimination of cholesterol? The answer is bile acids. If I ask you about bile acid sequestrants, make sure that you know that cholesterol is regulated by bile acid sequestrants. What is the function of cholesterol inside the body? It's a precursor for steroid hormones, and most importantly, it is the precursor for vitamin D. And why it is the precursor for vitamin D? Because vitamin D also has a steroid nucleus which is derived from cholesterol molecule. Cholesterol is the product of animal metabolism, because our body is not able to digest plant sterols, and that is one of the reasons often people who are suffering from hypercholesterolemia are suggested diets that are having increased amount of plant fiber. The predominant site of synthesis of cholesterol is the liver, but cholesterol synthesis occurs also in other tissues. So this is like a take-home message, and please focus on this slide because it will help you to answer some straightforward questions in an exam. Now, cholesterol is an amphipathic lipid. An amphipath is a molecule that has both hydrophobic and hydrophilic portions, and therefore, since it's an amphipathic lipid, it is a key component of cell membranes. And I told you, cholesterol through this particular hydroxyl group is able to bind to fatty acids through a ester bond, and that is the major storage form of cholesterol in the tissues. So which particular part of the cholesterol helps in storage in the human body? If I give you the structure of cholesterol and I ask you, you should be able to identify the hydroxyl group which forms the ester bond with fatty acids and facilitates its storage in the different tissues. In normal healthy adults, approximately 1 gram per day of cholesterol is synthesized, and if we follow the normal American diet, I'm sorry I do not have the data for the Middle Eastern diet, but for the American diet, an adult individual consumes around 0.3 grams per day of cholesterol. So that is the reason when people with hypercholesterolemia are treated, first they will try to limit the intake of cholesterol from the diet, and if that doesn't work, then they will inhibit the synthesis of cholesterol inside the body. However, a relatively constant level of cholesterol in the body, which is 150 to 200 milligrams per deciliter, is maintained primarily by controlling the level of de novo synthesis. And what I want you to do is to define for me what is de novo synthesis. De novo synthesis relates to the synthesis of cholesterol inside our body, and by the body is able to regulate the levels of cholesterol by controlling the synthesis of cholesterol under normal physiological conditions. Of course, people who suffer from hypercholesterolemia, which may be because of genetic reasons or maybe because of other factors, environmental reasons such as stress, the level of cholesterol's biosynthesis in these patients or individuals rises significantly, and the clinician has to then treat the individual with specific cholesterol lowering drugs or medication. We need to look at the synthesis of cholesterol, and as I said, it's a very complicated pathway, and I'm going to focus on very few steps. And what is the take-home message from here is that we are going to look at the rate limiting enzyme of cholesterol biosynthesis, because that is the target site for the action of the key cholesterol lowering drugs. So if I start the process, remember we talked about this when I mentioned ketone body synthesis. We have two acetyl coenzyme A molecules which condense to form acetoacetyl coenzyme A, and the enzyme that is catalyzing this process is thiolase. That's step 1. In step 2, acetoacetyl coenzyme A undergoes another condensation reaction in the presence of acetyl coenzyme A and water, same as ketone body formation, and you can see here you get beta hydroxy beta methyl glutaryl coenzyme A or HMG coenzyme A, and this particular process is catalyzed by HMG coenzyme A synthase, which is the rate limiting enzyme for ketone bodies but not cholesterol. Please put this down in your notebook. People often make this mistake. HMG coenzyme A is the rate limiting step for the formation of ketone bodies but not cholesterol. So once I have HMG coenzyme A, this particular HMG coenzyme A will undergo the first stage of cholesterol biosynthesis, where it will combine with acetyl coenzyme A, and this particular condensation that is happening between HMG coenzyme A and acetyl coenzyme A leads to the formation of mevalonate, which is a 6-carbon containing compound, and this particular step is catalyzed by HMG coenzyme A reductase. And in the process, because it's a reduction reaction, we require a proton, and that proton is donated by NADPH. This NADPH, of course, is coming from the pentose phosphate pathway, which we have talked about, and that tells you how pentose phosphate pathway helps in the synthesis of fatty acids, which we'll talk about, but also cholesterol. And the process is catalyzed by the enzyme HMG coenzyme A reductase. And this HMG coenzyme A reductase is the rate determining step or the rate limiting step in the process of cholesterol biosynthesis. This particular enzyme is under tight regulation by two molecules, one cholesterol, which is the end product of the pathway, and two by mevalonate. So both cholesterol and mevalonate, they inhibit this particular enzyme activity. Very, very important. This is the target site for a group of drugs which are known as statins, which are used for the regulation of cholesterol biosynthesis inside the body. So once I have mevalonate, it undergoes a certain series of reactions, predominantly a decarboxylation, and this leads to, since I am losing one carbon, I get C5 or a metabolite containing 5 carbon atoms, which are known as active isoprenoids. And then there are several condensation steps which I believe you will not need to know or even look at. But in through these condensation steps, we are able to convert isoprenoids to squalene, which is containing 30 carbon atoms. And in the process, we require NADPH, and this NADPH is again coming from pentose phosphate pathway. And squalene undergoes cyclization and again requires NADPH in the process, and is converted to a 30 carbon ring structure. And the ring, this particular 30 carbon atom containing molecule has 4 rings, which we looked at when we started looking at the structure of cholesterol, and that particular molecule is known as lanosterol. Again, there are several steps involved in this process, and I urge you not to go through the steps. Just remember that this particular reaction also requires NADPH, and any NADPH that is used in this particular process of cholesterol biosynthesis is derived from the pentose phosphate pathway. And lanosterol undergoes further metabolism through 19 different steps, and we get the 27 carbon atom containing cholesterol. Again, if you look at it here, you require NADPH, and this NADPH is coming from the pentose phosphate pathway. So the take-home message here is the rate limiting step, and the rate limiting step of cholesterol biosynthesis is HMG coenzyme A reductase, not synthase, so mark that. And I told you the regulators as well as why this particular enzyme is so important. So if I want to sum it up, what do you need to remember from cholesterol biosynthesis? And this is like the focus slide. All carbon atoms that are present, all the 27 carbon atoms that are present in cholesterol are derived from acetyl coenzyme A molecule. The synthesis of acetyl coenzyme A requires the presence of NADPH, and this NADPH comes from the pentose phosphate pathway. Apart from that, we also require ATP and oxygen. And this is my way of looking at cholesterol biosynthesis, so you will not find this in the book. There are 4 different steps. In the first step, we convert acetyl coenzyme A to HMG coenzyme A, and these steps are common to the ketone body formation steps. In the second step, we convert HMG coenzyme A to 5-carbon intermediate, which are called isoprenoids. And in the third step, we convert isoprenoids to squalene. And in the fourth step, we take squalene and oxygen and we form cholesterol. So these are the 4 very, very broad steps that are involved in the formation of cholesterol. You do not require to go through any of the individual steps or memorize any of the structures. Just remember that cholesterol has a steroid nucleus and it is derived from acetyl coenzyme A and requires NADPH and ATP and oxygen. Most importantly, NADPH that is required in the process of cholesterol biosynthesis is coming from the pentose phosphate pathway, and cholesterol is the precursor for steroid hormones and vitamin D. I think that is all that you need to know. And of course, as I mentioned, you need to remember, you need to know the rate limiting step, which is HMG coenzyme A reductase. And this particular enzyme is inhibited by a group of drugs which are known as statins. I think we already talked about these drugs when we talked about enzyme inhibition. There are different types of statin available, but statin are competitive inhibitors of the enzyme HMG coenzyme A reductase, and they are used for reduction of cholesterol and low density lipoprotein. There are pleiotropic activities of statins, which means there are activities other than their cholesterol lowering effect, and these pleiotropic activities are also very important. And the key pleiotropic activities are statins are anti-inflammatory in nature, so they are able to impair or impede the formation of atherosclerotic plaque. Statins help in increasing levels of high density lipoprotein cholesterol, and statins have other effects such as anticoagulant effect, so they are cardioprotective. I'm not going through all the pleiotropic activities. You can go through them in a typical review article detailing pleiotropic actions of statins, and I will upload a particular document where you can read these pleiotropic activities. Although we always discuss about the different beneficial activities of statins, the drugs have certain side effects. One of the side effects is myopathy and a particular very rare condition known as rhabdomyolysis. Also, statins should be administered with care because they increase certain levels of hepatic enzymes in the serum. So not all aspects of statins are good, but mostly statins are drugs that are widely used for the treatment of hypercholesterolemia. Of course, nowadays we have new drugs that have come up, and one of those drugs is the PCSK9 inhibitor, which are monoclonal antibodies inhibiting PCSK9, which help in the regulation of cholesterol levels as well. I believe this they should deal when they are teaching cardiovascular system, because these drugs were recently introduced in 2012 to be precise, and they have far better therapeutic efficiency compared to statins and have very less side effects. But they are extremely expensive. So that is what you need to know from the lectures on ketone bodies and cholesterol biosynthesis. And this particular part is important for short answer question. I will upload some questions for you to practice so that you are confident on this topic when you go for your in-course or your end-of-course assessment. So with that, I would like to end this lecture. Have a very nice day, and thank you for listening.